In this video, we're going to study the process of meiosis. In the previous lessons, you reviewed mitosis, which was a form of asexual reproduction where a parent cell, so let's say the parent cell starts off as a diploid cell, 2N, essentially duplicates everything, DNA and organelles, and then goes through this process where it divides into two identical daughter cells, and those daughter cells are identical to each other and to the original parent, and they also remain as diploid. And the purpose of mitosis, and especially as it pertains to humans, is to grow, to maintain the organism, to help repair the organism, to make identical copies of cells, to replace them in the event that they're damaged, for example, like when you get a scratch, and that wound heals over time. Now, the type of division we'll be studying in this presentation is something called meiosis, which is a type of division that involves forming gametes. So the reproductive cells like sperm and egg. So rather than occur in somatic cells like mitosis does, meiosis occurs in the germline cells, so the testicles for males and essentially the gonads altogether, so the ovaries for females. So what we're going to do is we're going to go through the entire process of meiosis, look at how, and at the end we're going to take a look at how it's different from the process of mitosis and the cell cycle that we saw earlier on. But this is the big idea. We start off with a parent cell. You'll notice that it's diploid. That parent cell will eventually split into two haploid cells, and those two haploid cells will split into four haploid cells called gametes. So already we can see that meiosis involves two divisions. Our first division is over here, and our second division is over here. And rather than end up with diploid cells like in mitosis, we end up with haploid cells, N instead of 2N. So you've seen this already, but the general idea is that in order to become you, you're a little bit older than this, but in order to become you, essentially what had to happen was that sperm fertilized the egg, that formed a zygote. The sperm was haploid, so was the egg. Egg from mother, sperm from dad. The zygote is diploid. And then over time, through mitosis, cell cycle mitosis, you had a bunch of these diploid cells, and they specialized and they became you. You're a diploid organism. So you can see that it's important that when a baby is being made, that the gametes are haploid. If the gametes were not haploid, let's say the gametes were diploid instead, which is wrong, so you know it's wrong, then you'd have an organism that would be tetraploid, and that may not develop properly. So the whole purpose of meiosis is to generate gametes that are haploid, so you can use those, combine them to produce a diploid organism, a diploid individual. So the general idea is that mitosis preserves ploidy, whereas meiosis would reduce ploidy. It divides ploidy in half. So rather than have diploid, we end up with haploid daughter cells. And we're going to see how that entire process works. So in humans, if you have 2N is equal to 46 in your cells, mitosis would produce cells that are 2N is equal to 46. But meiosis, that would produce cells that are N is equal to 23, haploid, half the number of chromosomes that you would have in your somatic cells. So once again, meiosis occurs in the sex organs in something called the germline cells, so ovaries for females and then testes for the males. The purpose of meiosis is to produce gametes, and then we use these gametes in sexual reproduction. So the sperm from one parent and the egg from the other parent combine, and we call that sexual reproduction because we have more than one parent involved in producing the individual. And that has several advantages that you already heard about, but one being that because we're getting genes from two different sources, we can create much more diversity in our populations. We can shuffle genes around a bit more and have different combinations instead of a bunch of clones that are created by means of asexual reproduction. And as you know, diversity is important for the health of a population. So just again to highlight what is happening and how all these different types of divisions fit together, what we're going to do is we're going to study the human life cycle. You can study this for a variety of things. We're going to focus on humans, which are similar to other animals. So the idea is that you have, let's say, two adults of reproductive age. These are multicellular organisms. They're diploid. You can see that with the 2n is equal to 46. And the arrows show the process. So these individuals in their germline cells are going to do the process of meiosis. And you know that meiosis doesn't preserve ploidy, but it halves ploidy. It cuts it in half. So if the adults had 2n is equal to 46, then their gametes will be n is equal to 23 after going through the process of meiosis. And you could essentially figure these numbers out for anything. If I had 2n is equal to 14, then my gametes would be n is equal to 7. Now, the next step is after the gametes are made is fertilization. The sperm combines with the egg, that's fertilization. N plus n gives you 2n. So we had n is equal to 23 from the egg, n is equal to 23 from the sperm. They come together to give you 2n is equal to 46. And then that organism goes through mitosis, and as you can see, the ploidy is preserved, right? The cells just divide over and over and over and over again, but they preserve ploidy because of that interphase that occurred in mitosis, which we saw earlier in the course. So that's in general the human life cycle. I could give you any life cycle, and you should be able to figure out the ploidy based on the process that's happening. So fertilization, you saw, essentially adds or doubles the ploidy. If you had n is equal to 23 and you combine two together, that's 2n is equal to 46. If I had n is equal to 7, n is equal to 7, and I combine those two together, it would be 2n is equal to 14. So the important point is looking at the process. Meiosis cuts the ploidy in half. Fertilization doubles the ploidy, and mitosis preserves it. So I'm just going to put times 1. Preserves it, keeps it the same, basically. So let's do another quick review of chromosomes, because those are super important when it comes to meiosis and mitosis. Remember that your human somatic cells, so anything but sex cells, they're diploid, and in humans it's 2n is equal to 46, whereas gametes are haploid, and in humans that would be 23, n is equal to 23. So 23 from the sperm and 23 from the egg. When they fertilize, they give you 2n, diploid, is equal to 46. So again, that's why it's important that your gametes are haploid, because in the end you need to be a diploid individual. If your gametes are not made haploid, then you will have more chromosomes than required, and that could lead to abnormalities, to developmental disorders, to organisms not actually developing at all, to miscarriages, so on and so forth, because you have too many of certain genes and too many of certain chromosomes. So what I want you to get is, we'll take a quick look at this picture here. This is called a karyotype. A karyotype is just a picture of your chromosomes. And specifically, this is a picture of chromosomes in a somatic cell of a human, and I know that because if you look at it carefully, you have 23 pairs of chromosomes. 23 pairs, which means 46 chromosomes. And so what I want you to get is that in each pair, one comes from mom, and the other comes from dad. Mom, dad. Mom, dad. Mom, dad. So you have 23 pairs. 22 pairs are called autosomes, and they're chromosomes that contain DNA with genes that control basically everything except for your sex. So genes that might control your hair color, your eye color, height, and various things. And this is simplified, but you also have a pair of sex chromosomes that determine your sex, if you're a boy or a girl. So if you have XY, that means you will become a boy, and if you have XX, you will be a girl. And that's because the Y chromosome has a gene that essentially triggers your development into a boy. So the idea is that only men, boys, can give away a Y chromosome. So here we have one from mom, one from dad. Mom, dad. So dad could give an X chromosome or a Y chromosome. It's a 50-50 chance. If the father gives X, then the individual will develop into a girl, and if the father gives Y, then the individual will develop into a boy. A mother can only give X because she only has X chromosomes as sex chromosomes. So again, autosomes are the chromosomes that control everything but sex, and that's simplified. And then your sex chromosomes are the chromosomes that control your sex. But there's other genes on there in reality. It's just that we simplify it and say that they only control your sex, but there's much more on there than just that. So over here we can see karyotypes. We can see a karyotype for a female and a karyotype for a male. You'll notice there's 46 chromosomes, so I know it's from a somatic cell. There's 44 autosomes, or 22 pairs of autosomes. And then there is one pair of sex chromosomes in each of the karyotypes. You'll notice we have XX over here, so that means we have a girl, and XY over here, that means we have a boy. And again, these pairs, one is from mom, one is from dad. Mom, dad. Mom, dad. We're going to talk about these pairs in a bit more detail in just a few minutes. So those pairs that you saw in the previous slide, we call them homologous pairs. The chromosomes that pair up, when mom and dad give you their chromosomes will pair up together based on similarities. And so those similarities include things like the length of the chromosome. You'll notice that these two chromosomes have the same length. Another thing is the genes on the chromosome. They actually have the same genes on them. So for example, this band over here represents a gene. It might be a gene for, I don't know, eye color. So let's call it EC. This one was from dad. This chromosome here is from dad. Now on the homologous chromosome, the partner chromosome from mom, this gene over here is also a gene for eye color. This might be a gene for, I don't know, hair color. And on the same location, but on mom's, there's also a gene for hair color here. So in addition to having the same length, these homologous chromosomes also have the same location of their genes. And remember, a location of a gene is called a locus. Now, one thing I do want to emphasize here is that even though this is the eye color gene and eye color gene, you could have a different version of the eye color gene from dad and a different version from mom. So for example, dad might have given you the brown version of the eye color gene, and mom might have given you the blue one. And then somehow your body knows, or your body figures out, that only one of these is shown in you. So in this case, usually it's the brown color that dominates the blue color. But the idea is that they're both eye color genes. Different versions of the same gene, that's called allele. So for example, there's the eye color gene, but you could have the brown allele, or you could have the blue allele. There's a hair color gene. You could have a red allele or a brown allele. That's simplified, but that's a general idea. Again, here's another pair of homologous chromosomes. One is from dad, from the sperm, one is from mom. You'll notice same length. You'll notice same genes and gene location. There might be different versions of the gene on it, but same gene and gene location can be found on it. The other similarity that homologous chromosomes have is that they have the same centromere position. So, for example, you might have a centromere over here from the chromosome that comes from dad. On the same relative location, you have a centromere over here from the chromosome that comes from mom. So here's just another illustration of homologous chromosomes, the ones that pair up together that you saw in the karyotype. This is my father over here, and this is my mother over here. Here we have the homologous chromosomes, you'll notice they have the same length, same centromere position, and same genes over here, and same location for those genes. Now again, they could have different versions of the gene, but the idea is that it's the same location for whatever controls. So, for example, the green over here might control for height. So the green over here would control for height. The orange over here might control for eye color. This would control for eye color over here. This might control for hair color. This would control for hair color over here. This might control for a certain disease, and this would also control for that same disease. But I might have different versions of that disease gene, or different version of the eye color gene. And again, those different versions of the same gene are called alleles. And so, depending on what versions you get, you might have different characteristics. So if you get the brown eye allele and the blue eye allele, the brown one would show up. But if you get two blue ones, one blue one from mom, one blue one from dad, two blue alleles, then you'll actually have blue eyes. And we'll learn more about that we'll learn about that in more detail later on, but that's the general idea behind how certain characteristics show up in you. So there's a little bit of an exception to the homologous chromosome rules. Remember I said homologous chromosomes need to have the same length, same gene location, same genes. They can have different versions, but same genes, and same centromere position. So here we can see the sex chromosomes. You can see two X chromosomes. They're fine. They follow that general idea of length, gene, and centromere position. But the main exception is the sex chromosomes in a male. The XY chromosome, they are homologous, even though they're not the same length. And they don't have the same genes on them. They're still considered homologous. They do have some genes in common, which is why they can kind of meet each other and pair up and realize they're similar, but that's the major exception there. So you'll notice there is quite a big difference in terms of the size when you're looking at the X versus a Y chromosome, and you can probably already guess which chromosome has more genes on it. If you want to take a guess before I tell you. That's right, the X chromosome. Why? Because it's a lot bigger and it has a lot more room to carry that DNA with all the little nucleotides on it and that code as well. Now just to let you know, the Y chromosome has a gene called the SORI gene. It's basically a gene that triggers the development of male characteristics in embryos as they start to develop. Now in humans, sex determination is determined by your combination of XY chromosomes. So females are XX, whereas males are XY. But other animals have different forms of sex determination, and I recommend that you read up on it just to have a general idea. Just to give you an example here, if you look at, let's say, bees. Bees, if the individual is diploid, it'll become a female. But if the individual is haploid, it'll become a male. You'll also notice sometimes sex determination is based on temperature. So if the eggs are kept warm, the eggs become females. If the eggs are kept cool, well, most of the eggs become males. So there's different types of sex determination. I do want you to read up on them to have a good idea of some examples that you could maybe provide, and realize that, you know, even though we study humans primarily in this course, at least in this first unit, there's many other methods of sex determination and many other ways that genes are used to determine sex in nature. So here we can see a summary of everything we've spoken about so far. We have our fertilization event, where you have two haploid cells combining together to give you a diploid cell, and that diploid cell will eventually become a multicellular organism if we're talking about humans. You can see that after fertilization, you have the chromosomes from dad and the chromosomes from mom in the same nucleus now, and then you'd have homologous that would pair up. So this one and this one would kind of pair up together eventually, and this one would kind of pair up together eventually. Homologous chromosomes, again, should be the same length, same gene, same centromere position, with the exception of the X and Y. They do have some regions in common with each other, So that's why they do pair up, but not all the regions are shared in common with each other. So now we're going to go into our details of meiosis. Meiosis, as we study it, is the process of making gametes, sperm and egg. And there's three main steps to meiosis. There's an interphase step that you can see over here. And that's where we essentially, just like the cell cycle mitosis, there's preparation for division. We double things, we double DNA, we double certain organelles, and we prepare a variety of other things. And then we get into our other two parts called meiosis 1 and meiosis 2. So meiosis 1 is over here. We sometimes call meiosis 1 the reduction division because it takes a diploid cell and produces two haploid cells. Now you'll notice those two haploid cells, so let's just take a look at our chromosomes here. So at the beginning our cell had 4 chromosomes, 4 of these little lines here. After interphase it still had 4 chromosomes, but they were replicated chromosomes. After meiosis 1, now each cell has 2 chromosomes. So compared to the initial parent, which had 4 chromosomes, these guys have 2 chromosomes, so they're haploid, but the problem is their chromosomes are still, you'll notice, replicated chromosomes. So there needs to be another division that will divide those replicated chromosomes into non-replicated chromosomes, and that's meiosis 2. So now there's another division for each of these cells, and now you'll notice that we have our non-replicated chromosomes, 2 chromosomes each, and they're haploid, 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 haploid. So the end result of meiosis is you get 4 gametes that are produced. So we have our original cell, interphase happens, doubles our DNA. Meiosis 1 makes the haploid cells, but the chromosomes are still replicated chromosomes or duplicated chromosomes, so we got to make sure we take care of that. So we do another division. That's meiosis 2 over here. And that gives us our haploid cells with 2 chromosomes each, which is half of what the original parent had. It had 4 chromosomes, and now these have 2 each. And as a result, they are haploid. So we're going to go and take a look at all the details that are involved in this. These are the big ideas, the general process, and now we're going to take a look at the details involved and the major steps. So one more thing to mention is that if we're looking at mitosis cell cycle, it really only has one division. It has its interphase that happens, so it would look kind of like this. So if this, let's say we had the same cell going through mitosis, we'd have our 4 chromosomes. We'd have our interphase happening. So we'd have our 4 chromosomes, and now they're replicated. Now I'm not making them different sizes and just showing 4 replicated chromosomes. And then we'd have our division with our 4 non-replicated chromosomes within. So the number of chromosomes and DNA stays the same as the original parent, just like that. And it was only one division involved. One division was involved in this process. That's just a summary of what I just explained in the previous slide. So mitosis, 1 division, and our ploidy is preserved. Meiosis, 2 divisions. So this is meiosis number 1, meiosis number 2. And our ploidy is halved. We get 4 haploid cells, whereas in mitosis we get 2 diploid cells. So 2 cells versus 4 cells, diploid versus haploid, 1 division versus 2 divisions. So you can see that there are several differences that exist already just in the big ideas of mitosis versus meiosis. So let's start off with interphase. Again, interphase, just like we saw with the cell cycle, is preparation for division. So the cell is preparing for meiosis 1. You can see, similar to mitosis, we have some chromatin rather than chromosomes. We have our centrosomes that were duplicated, so there should be 2 centrosomes, and those again are going to shoot the spindle fibers out to attach to chromosomes eventually, just not yet, but soon. Our DNA is going to be duplicated, so there is an S phase in this meiosis process. So for example, if you had a cell that was 2n is equal to 6, it would start off with 6 DNA molecules, and after replication, it has 12 DNA molecules. 12 DNA molecules. And again, you can see spindle fibers are starting to form and radiate, and then eventually, or right now at least, you can see that there's still a nuclear membrane, also sometimes called a nuclear envelope, and you can still see the nucleolus. But the main idea is that our DNA is duplicated, our centrosomes have been duplicated, and our DNA is still in the form of chromatin right now. So it's preparation for meiosis 1. So now we get into meiosis 1. Meiosis 1 has several parts to it. The beautiful thing is the name is similar to mitosis. There's prophase, there's metaphase, there's anaphase, there's a telophase, and the main difference is that they're designated with a 1 to show that they're part of meiosis 1. So there's prophase 1, there's metaphase 1, so on and so forth. So let's take a look at what's happened so far. You can see that in meiosis 1, prophase 1, the nuclear membrane has broken down. You can see that the centrosomes are starting to move away from each other, and spindle fibers are coming out of them. You can see that our chromatin has become chromosomes. Remember, we had 2n is equal to 6, that means we had 6 chromosomes. But here's what I want you to get. In mitosis, the chromosomes didn't really pair up. You'll notice in meiosis, though, the chromosomes, the homologous ones, pair up with each other. They connect to each other. So all these homologous ones, so the 2 long ones connected to each other, the 2 short ones and the 2 medium ones, they all connected with each other. When the homologous chromosomes are paired up, we call that a tetrad. Tetra meaning 4. What is there 4 of? Well, there's 4 chromatids. So this and this, these are sister chromatids. And then the red one has sister chromatids too. So since there's 4 chromatids, we call that a tetrad. So in mitosis, cell cycle, prophase, there's no pairing up of homologous chromosomes. The chromosomes don't really care about each other that much. They just kind of eventually line up on their own. But here in meiosis prophase 1, the homologous chromosomes have paired up towards each other. Now, another thing that's going to happen during this phase is something called crossing over, and we're going to see what that means in the next few slides. But again, nuclear envelope disintegrates, nucleolus is gone, our spindle fibers are coming out of the centrosomes, and the centrosomes are moving to the poles. Even though you don't really see it here, the spindle fibers will attach to the chromosomes at their centromere, and that's usually in late prophase 1. So eventually, spindle fibers will attach kind of over here and over here. And then same thing over here and over here and over here and over here. So in this picture right now, we have 1, 2, 3, 4, 5, 6 chromosomes. And if I were to ask you how many tetrads there are, you would say there are 3 tetrads. 3 tetrads that's occurring here. Now you'll also notice something else that's important. There's color coding happening here. And the color coding just tells you which chromosome is the chromosome that came from dad and which one was the one that came from mom to begin with. So let's just say that red is from mom, maternal, and blue is from dad, paternal. I mean, it's not the reality, but it doesn't really matter. Blue's from dad, red's from mom. So let's talk about the idea of crossing over. Crossing over is the process that occurs during prophase 1 of meiosis 1. You can see that we have our homologous chromosomes over here, and they connect together. And what they do, the reason they're connecting together is because they want to exchange some genetic material. At the parts where they're touching here, they're going to swap genetic material. So you'll notice some of the red went to the blue, and some of the blue went to the red. This doesn't happen in mitosis. You don't have a changing and a swapping of genetic material. But in meiosis, you do. And this is one of the things that leads to even more diversity in your gametes. You're reshuffling the genes, not just with the fertilization of sperm and egg, but you're also reshuffling what your chromosomes have. You're getting different combinations and flavors here, which is super, super important and advantageous for the diversity in populations. So the sites where the chromosomes kind of connect to each other and attach very closely to each other. Those are called the chiasma or chiasmata. The actual attachment is called synapsis, and the actual exchange of the information at the chiasmata is called the crossing over. So only homologous chromosomes, though, will do crossing over with each other because they can actually attach to each other. Chromosomes have to be similar enough to recognize each other, to connect to each other, to synapse, and then to cross over afterwards. So again, it leads to much more diversity, and it happens between homologous chromosomes. Okay, so the homologous chromosomes, these are non-sister chromatids. This is not a sister chromatid to this one, right? Remember, sister chromatids are on the same one. So this happens between homologous chromosomes. Now in reality, I mean, in this picture it's a bit simplified. We're just showing crossing over happening at one point. Crossing over actually happens at several chiasmata on the chromosome. So you'll notice there's a crossing over point here, a crossing over point here. So several regions can have crossing over occur, and therefore that leads to even more diversity in that chromosome. So again, this does not happen in mitosis. It only happens in meiosis. So in mitosis, the sister chromatids of a given chromosome are always identical to each other, but in meiosis, the sister chromatids are no longer identical after that crossing over happens, right? You'll notice that the sister chromatids, this one's all red DNA, and this one is red, and then some green. So it's no longer identical to its sister chromatid that it was attached to before. Same idea over here. So now the sister chromatids are no longer identical to each other. This idea of exchanging genes and getting new combinations is called recombination or mixing. So it's called the recombination of genetic material. Recombination. Sometimes you'll hear it called genetic recombination. And again, just to show you that this idea of crossing over actually occurs at several spots, not just at one spot on the chromosome. So after all that magic happens in prophase 1, we then move on to metaphase 1, where the spindle fibers line up the pairs along the metaphase plate. So in mitosis, the chromosomes weren't paired up, they were lined up single file. Whereas in metaphase 1, it's the homologous pairs that are lined up. So they're lined up pair by pair by pair instead of single file, chromosome by chromosome by chromosome by chromosome by chromosome by chromosome. Although this picture doesn't show it, please do realize that the sister chromatids are no longer identical due to crossing over. So just remember that as you go through this presentation. You can sort of see that here, but it's not very apparent. So again, the idea is that the chromosomes are not single file, but rather the homologous pairs are lined up along the metaphase plate. During anaphase 1, the spindle fibers shorten, the cell elongates to make that oval shape, and you'll notice that rather than splitting apart sister chromatids like in mitosis, we're just separating the actual homologous chromosome pairs from each other. So this pair is going towards the pole, this pair is going towards a pole. So is that pair, this chromosome from the pair is going towards a pole, and this chromosome from the pair is going towards this pole. So is this chromosome from the pair, and so is this chromosome from the pair. So the idea is that by the end of this process, you have 3 chromosomes on one side and 3 chromosomes on the other side. They're still replicated chromosomes, they're still duplicated because we didn't separate the sister chromatids. We separated the homologs from each other. So the homologs were separated from each other. And so that gives us a term called the law of segregation. And what that means is that the homologs of a pair separate from each other during gamete formation. So in the gametes, you're not going to have the homologous pairs together anymore. You'll only have one from the pair in each gamete. So that's called law of segregation, meaning we separate the pairs from each other. You can think of it kind of like in a class where you have maybe friends that keep talking to each other. the teacher might have to separate those friends because they don't pay attention, and that would be segregating them, separating them from each other. That's the law of segregation. So from now on, the teacher says, you know what? In any group that you have to work in, you guys can't work together. So you're going to have to be separated. You won't be together anymore. That's exactly what's happening here. So they were too close to each other, and so we're separating them and putting them in different cells. And that's called the law of segregation. Remember that. It's super important, especially when we study Mendelian genetics in more detail. So what I want you to get here is that we still have 6 chromosomes so far. We still have 12 DNA molecules. I want you to see that the sister chromatids are not identical to each other. That's great. That means good diversity for gametes to create a whole bunch of different types of organisms. And I also want you to get that there's no more tetrads. The tetrads have been pulled apart, right? You separate the homologs. So there's no more tetrads in this picture. So after anaphase 1, we go into telophase 1 and cytokinesis. So cytokinesis, you can see the cleavage furrow again. The cell's going to split into 2 cells. The cells this time will not be identical to each other because of that crossing over that happened, right? You'll notice that they don't have the same color of chromosomes anymore. And you'll also notice that the cells won't have 6 chromosomes anymore. They'll actually have 3 chromosomes each. There's 1, 2, 3, once that's split off, and then 1, 2, 3, once that's split off. So in the end, we have 2 haploid cells, n is equal to 3, n is equal to 3, but the chromosomes are still replicated, so we still have to go through another division phase. But essentially, that's the end of meiosis 1. So we have our telophase happening, and sometimes the chromosomes become chromatin again, and then they recondense and go back into meiosis 2, and other times they don't become chromatin again, so they can just go straight into meiosis 2. But the idea is that there's usually no interphase 2. There's no interphase between meiosis 1 and meiosis 2. So for the purpose of this course, we're going to go with, after meiosis 1, those 2 cells that are produced, they're going to go straight into meiosis 2. There's no more doubling of DNA that's happening. We just really need to chop down these chromosomes so they're now unreplicated chromosomes. That's the whole purpose of meiosis 2. Meiosis 1 was reduction to get diploid to become haploid. Meiosis 2 is going to be to take those replicated chromosomes, make them unreplicated chromosomes. Sometimes you might see a nucleus reform around each set of chromosomes, but before meiosis 2 happens, that nucleus has to be disintegrated again, and then it can go on and proceed. So right now what we're going to do is we're going to go take a look at meiosis 2. We've seen interphase, we've seen meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Now we're going to jump into meiosis 2. So here we can see that our cells are entering prophase 2 of meiosis 2. You'll have the centrosomes move towards opposite sides of the cell. Spindle fibers will attach to the centromere of the chromosomes. There's no pairing up this time because there's no homologs. The homologous chromosomes have been separated from each other, so no pairing up. If a nuclear membrane did reform while the nuclear membrane was disintegrated, the centrosomes had to be replicated again, but there's no replication of DNA or anything like that that occurred. The spindle fibers, like I said, will attach to the centromeres of each chromosome, but there's no pairing up this time because, again, there's no homologs. Okay? So what I want you to get is that in prophase 2, we're following the same events for 2 cells because 2 cells were created from meiosis 1. So metaphase 2, the spindle fibers will move the chromosomes to the metaphase plate, to the middle of the cell, and this time they're in single file. This is similar to mitosis. There's no pairing. The main difference, though, you'll notice is that in mitosis, the chromosomes, the sister chromatids of the chromosomes would have been identical. But because they're crossing over, the sister chromatids are no longer identical to each other. So even though it's very similar to mitosis, there's still some differences. The other difference too is that, well, this is now a haploid cell, and so is this, whereas in mitosis you'd be dealing with a diploid cell. Then we have anaphase 2, where the spindle fibers shorten and separate the sister chromatids this time. So rather than separate homologs, because homologs are not in the same cell, we're separating sister chromatids, similar to mitosis. But again, the main difference being that this is haploid cell, and the sister chromatids are not identical to each other, right? Because of that crossing over that had happened in meiosis 1, prophase 1. So we can see here we still have 6 DNA molecules, 6 DNA molecules, 6 chromosomes, 6 chromosomes. Then we get to telophase 2 in cytokinesis. So essentially, by the end of it, we have 2 haploid cells, n is equal to 3, with unreplicated chromosomes. And the haploid daughter cells are different from each other. They're not identical to each other because of that crossing over that happened. They're not identical to the mother, to the parent cell anymore, because of the crossing over, because they're haploid. So there's lots of diversity here that can be used. There's lots of diversity in gametes, which can be used to create many diverse offspring or kids. But the idea is that you'll have a nucleus that reforms, nucleolus will reform, spindle fibers are gone, and eventually the cells will split completely to get your 4 haploid daughter cells that have half the number of chromosomes and half the number of DNA. So in a way you can think of it as meiosis 1 chopped the number of chromosomes in half, whereas meiosis 2 completely chopped the amount of DNA in half compared to the original mother. So for example, if you had, let's say, 46 chromosomes, there's an interphase that happens to give you 92 chromosomes. And then the 2 divisions happen during meiosis 1 to get you back to 46. And now, what I should have been saying was not chromosomes of DNA. If you had 46 DNA, interphase happens, you get 92 DNA. And then after the first division, you go back to 46 DNA and 46 DNA. Now I know it looks like nothing happened, but in reality something did. What we did was these 46 DNA molecules were found on 46 chromosomes. Right? They were non-replicated. When we did replication, those 92 DNA molecules were still found in 46 chromosomes. Now, these 46 DNA molecules are found on 23 chromosomes each. So we chopped the number of chromosomes in half. But we still have 46 DNA molecules because those chromosomes are still replicated. So what we have to do is we do another division to get us down to 23 DNA per cell. 23 DNA per cell on 23 chromosomes. So our chromosomes are halved compared to the mother, and our DNA is halved compared to the mother or to the parent cell. So here you can see the entire process. I recommend that when you're studying meiosis, you start off by looking at the process holistically. So all the steps together, because it's a nice, easy way to embark into the journey of understanding meiosis. You can see how each step goes into the other. And you can also analyze the key terms, the key words, all in one area. So what I kind of explained earlier was about that replication that happens. So in meiosis, there's still a replication that occurs. And a lot of students wonder why. Why do you have to do a replication at all? Well, if you think about it, if you didn't do that replication of DNA, let's say you had 2 DNA and there were 2 divisions, there was no interphase for replication, well, the first division would give you 1 DNA, and then the second division would give you 0.5 DNA. So you'd have a quarter of the DNA that you started off with. So the idea is that if you let's say start off with 4 DNA, you double it first to get 8 DNA. First division happens, you get 4 DNA. So we're back to the same amount of DNA. But the thing is that we have half the chromosomes, right? These 4 DNA were on 4 chromosomes to begin with. This 8 DNA is on 4 replicated chromosomes. And this 4 DNA will be on 2 replicated chromosomes now, because it's split. Now we need to do meiosis 2 to get down to 2 DNA. And that 2 DNA is going to be on 2 chromosomes each. And so you can see that in this picture form over here. Here we have 2 chromosomes, 2 DNA. Here we still have 2 chromosomes, but 4 DNA because of replication. After meiosis 1, we have 1 chromosome, but 2 DNA because it's a replicated chromosome. So we're down to haploid in terms of chromosome number. So 1 chromosome, but still 2 DNA because it's a replicated chromosome. After a second division, we have 1 chromosome, 1 DNA. And that's the same in each of these cells. Now this picture is a bit simplistic. It doesn't show that there was crossing over. In reality, this would probably be like a red color here. And this one here would have a little blue color on it because of crossing over. So sometimes pictures are a bit simplified. They don't show all the steps involved, but that's what you would see. This here is just a summary of the entire process that we just visited together. Another summary picture. The next few slides will be animations of the process of meiosis. So I'll briefly walk you through what the animation is showing. To view it, you need to make sure that you're in the presentation viewing mode. So that previous slide was the interphase. Here we can see the prophase 1 with the pairing of the homologous chromosomes. There should be crossing over that occurs, so there would be an exchange between the orange and the blue chromosome. Here you can see the metaphase 1 of meiosis 1. The homologs are lined up in the middle of the cell. And in reality, again, there should have been some exchange to show crossing over, so there'd be a little bit of blue on here and a little bit of orange. On here. Anaphase 1 will occur, and the homologs have been split. There will be a telophase and cytokinesis to get 2 cells now. Over here you're going to see that the spindle fibers have attached to the chromosome within each cell, have brought it to the middle for metaphase 2. Anaphase 2 will occur by splitting the sister chromosomes apart, and then you'll have a telophase 2 cytokinesis to get your 4 haploid cells with 1 chromosome and 1 DNA each, which is half of everything from the mother. So the parent cell had 2 DNA, 2 chromosome. Now we have 1 DNA, 1 chromosome in each haploid cell. Keeping in mind that there would be some crossing over that occurred to get some diversity in the chromosomes that you see. So they wouldn't be necessarily identical to each other as you see there. There are some great videos for you to watch to reinforce your knowledge of meiosis and to compare meiosis to mitosis. Comparing mitosis to meiosis is a very important aspect of this course. You should know the major differences. You should know, for example, the major differences in terms of the big ideas. For example, mitosis starts off with a cell that's 2N and ends off with 2 cells that are 2N that are identical to each other and also identical to the parent. Whereas meiosis starts off with a, this is a parent here, sorry, 2N, let's go with 2N over here. Meiosis starts off with a 2N and ends up with 4 cells that are not identical to each other, and they're also not identical to the parent. There are many other differences. So you can see, for example, metaphase of mitosis versus metaphase 1 of mitosis. You have single file pairing instead of pairing a single file metaphase instead of pairing of the homologous chromosomes like you would have in metaphase 1. In prophase of mitosis, the homologs don't pair up, whereas in prophase 1 of meiosis 1, the homologs do pair up and crossing over happens. There's no crossing over for mitosis because we want to produce clones. By the time we get to anaphase 1, telophase 1, and get into meiosis, we have haploid cells now in meiosis 2. Whereas in mitosis, there's only one division and it ends up in diploid cells. But again, in meiosis, we have haploid cells now. There are some similarities between meiosis 2 and mitosis, but keep in mind that sister chromatids are no longer identical. We're dealing with haploid cells in meiosis 2, not diploid cells. So there are major differences as well. You can't say that meiosis 2 is just like mitosis, but there are some similarities, and it would be good for you to know what those similarities are. Here we can see a summary table that goes over the major differences between mitosis and meiosis. This is not exhaustive. There's more that you can find out there, and it's always good to do some reading and look at textbooks and try to analyze some more of those differences. But just a few basic differences: the number of divisions. In mitosis there's 1, in meiosis there's 2. Number of daughter cells: 2 for mitosis, 4 for meiosis. Daughter cell conditions: are they diploid or are they haploid? Well, mitosis makes diploid cells. They have the same number of chromosomes in DNA, whereas meiosis makes haploid cells. They have half the DNA of half chromosome number. How do the daughter cells compare to the parent? Well, for mitosis they're identical, but for meiosis they're haploid and their DNA has been shuffled by crossing over. When does synapsis happen? When does crossing over happen? Well, it doesn't happen in mitosis, never. There's no tetrads. Whereas for meiosis it happens in prophase 1. Sister chromatid comparison, well, they're always identical in mitosis, but because of crossing over, sister chromatids are not identical in the meiosis process. Chromosome alignment, well, they're always single file in mitosis. They can be single file in meiosis, but only in meiosis 2. But in metaphase 1 of meiosis 1, the homologs are paired to each other. Our main purposes are different too. For mitosis, it's mostly growth, repair, and maintenance. Some organisms might use it for reproduction, but for us, it's these main reasons here for mitosis. Whereas meiosis, it's for gamete production. Another name for gamete production is gametogenesis, meaning genesis to create gametes. Genesis means to create, gameto, gametes. And then interphase. Well, yeah, interphase does happen in mitosis. It happens before prophase. And it also happens in meiosis, actually, so it's a similarity, except that it happens before prophase 1. Another difference you can take a look at is where they take place and when they take place. So mitosis takes place in the somatic cells, whereas meiosis takes place in reproductive cells, ovaries and testes. When? Well, mitosis happens kind of all the time throughout life, whereas meiosis happens at sexual maturity, when the organism is old enough to be able to produce these gametes. So here are some slide questions. There'll also be some worksheets for you to attempt. It's always good for you to try to draw these out, draw the steps out so you can have a good understanding of meiosis rather than just memorize it. So practice drawing them out, practice answering these questions, and practice making tables where you can compare mitosis and meiosis to each other. You can see that there's some good readings you can do. So page 152 to 159 covers the concept of mitosis and meiosis. There are some great questions you can practice with on page 160, and it's always great to practice with the worksheets and the slide questions that you've been provided with. There will be another presentation to accompany this one that goes over the diversity that meiosis can create in terms of the gametes and how that's good for populations. So keep an eye out for that, and do make sure you watch that as well.